Okay, let's get started. Howdy. Okay, so welcome to the second week of ESENS 766. So in the previous lecture, we talked about the chemistry and biology underlying the biomolecules that carry information and whose data need to be learned, right? So that's what bioinformatics is about. So I think so far you should have a better idea about the chemical basis of these molecules, right? So the reason we are talking about this is the following. Now, why do we think that the DNAs, RNAs, and proteins can be represented in one-dimensional text? The fundamental reason is that they're all biopolymers. They're all like repeating units, right? Nucleotides or amino acids linked together in a chain, right? And what links them together? The covalent bonds, right? So in other words, if you were to use a language model to learn the text written in the protein language, right? Or nucleotide or RNA language, right? What you will not learn is chemistry. You will not learn chemistry out of it. Because you can have any kind of bonds, covalent bonds between any kind of neighboring amino acids or nucleotides, right? Then if that is the case, what are we learning? By using, for instance, a language model on protein sequence data, what are we learning? If it's not chemistry.
Just a distribution for the next protein.
The distribution, what determines the distribution if it's not chemistry? What determines the distribution? Good point.
Based on our training data, will be like the highest probability for the next one.
Right. So what is underlying such distribution, which is to be learned by any kind of model? Underlying rule is not chemistry, as we said that, any kind of covalent bonds, right, can link any kind of pairs of nucleotides or amino acids. And that means what you are learning is sort of, the biological distribution. Is that right? Because all the sequences in your training sets are protein sequences that have been sampled by nature, right? So how did nature, out of all these 20 to the power of L, where L is the length of the protein length, out of all these, astronomical number of protein sequences, only some of them are sampled, right? So in other words, you can imagine there is a fitness landscape of all these protein universe, and then somehow what we have seen, right, in the training data are probably the sequences that have been evolved and survived so far, right? So something to keep in mind. Whenever you do data science, you first have to ask yourself what data, what is behind the data, okay? Because at best, you can mine from the data is the information encoded in the data. So if you know what to dig, then it might help you to come up with a better model to dig such information, okay? And we also said that these texts are actually, ordered, right? So some alphabets, human language, are written left to right or right to left. Here we say there is an order. Why? Well, the order is largely because of the chemistry. Again, in the case of the in the case of the RNAs and DNAs, right? The order is going from the 5' end to the 3' end, right? If you remember, there's a central sugar, right? Called deoxyribose or ribose, right? Then there's, if you go around that 5-membered sugar, there are 3' and 5' position, right? 5' is trying to grab the phosphate group, right? The 3' is OH group, right? Which is connected to the next phosphate, right? In the case of the proteins, you go from the N-terminus to the C-terminus because, in each amino acid, there is a central carbon, right? And then, it's connected to 4 partners, right? 2 of them are amino group and a carboxyl group, right? And the fourth one is always a hydrogen. And the third one is the one that differs the 20 different amino acids. That's the side chain, right? So there is an order as well going from the N to C terminus, right? I'm saying this is because even as recent as our own language models research lately, sometimes we had to use the forward and backward models to learn both the N to C and C to N. Especially if you are familiar with autoregressive language models, which are modeling the distribution of xi, where xi is the ith positions of a letter, given x1, x2, all the way to xi minus 1, right? Given all the previous positions, choices. So that's an autoregressive model. So in that case, the order matters, but biologically you think you would rather go from N to C alone, but empirically, because of the limitation of the model, sometimes you may find it's also useful to go from the other direction and use both models. So certain things to think about. We also talked about the four levels of protein structures. At the primary level, it's actually just the protein sequence, right? So it's like a beads, amino acids threaded together, right? And there are 20 different colors of the beads, and some of them are positively charged, some are negatively charged, some are not charged but polar, and some are hydrophobic who like to mingle with each other and hide from water. So they usually hide in the core of the protein. Because of all these different characters of amino acids, because of the non-bonded interactions between them, together with the covalent bonds that thread them into one chain, these proteins actually fold into a 3D structure, right? So locally you have these alpha helix and beta sheets, which are local structures. I didn't explain what they mean exactly, so that's part of the concept today. And then globally, these secondary structures are going to pack together into a global tertiary structure. So usually when we talk about protein structure prediction, we're talking about tertiary structure. And we call the primary structure the sequence, okay? The multiple chains or multiple proteins may interact with each other. In this case, 2 copies of molecule A and 2 copies of molecule B come together to form a hemoglobin that carries oxygen in your blood, okay? So, and then similarly, there are secondary structures for RNAs as well, so I'm not giving more details into it. But pay attention that unlike proteins where you have the non-bonded interactions like van der Waals, electrostatics, hydrogen bonds, and so on, in the case of the RNA or DNA, I said that the RNA double helix, this double helix, why you have a twisted double helix, that's because of the hydrogen bonds between the nitrogenesis bases across the chains, right? So, but the RNA is usually a single chain, but then they form a very sophisticated secondary structures, again because of the hydrogen bonds between these nitrogenesis bases. In other words, we call them base pairing. A with T, C with G. So in other words, it's not a surprise at all that one of the earliest biomolecule text generation or generated models to design molecules first occurred with RNAs, okay? Because they are a little bit more structured and patterned because there's a very strong base pairing pattern that determines the underlying secondary structure, whereas in the case of the proteins, not only they are much longer, but also the non-bonded interactions are much more sophisticated than the base pairing pattern in the RNAs. Okay, that's something to pay attention to. And I just said that because as you go from the sequence all the way to individual tertiary structure or the multiple proteins quaternary structure, the experimental technology's throughput is just decreasing exponentially. So that's why there is a goal of starting from the sequence and predicting the structure of individual proteins. That was a breakthrough made by AlphaFold last year. And they are now trying to solve this. They already have one paper already in bioRxiv predicting the complex protein-protein complex structures, okay? But inversely, you can also say that, well, in this case, you can only tell me to predict how a nature sampled sequence may fold into a three-dimensional structure or interact with its partner in three-dimensional space. What about those large space of sequence universe that hasn't been sampled by nature yet? So inversely, we have the design task that you may want to start with the ideal function or structure of a protein, because we said a naive assumption is that the shape or the structure of the protein determines its function. So if I say that I need a protein that can do this function extremely well and that function can be encoded by this shape, then I would start with this shape and try to design a sequence that optimally adopts that shape, right? So that's a design question, that's an inverse question, right? Or in the case of the drug design, you want actually your drug molecule to bind a target molecule in the human body very well, or bind to the virus, like the COVID-19 spike protein, right? You want antibodies that can neutralize, right, those virus proteins. In those cases, you start with a protein target in human or in virus, and you try to design some sequence or small molecule formula that can optimally bind or neutralize those targets. Okay, so these are the questions. Some of them will be addressed, but I will focus mostly on structure prediction and design, and use these as focal examples to show you how we address, don't forget our goal, multimodal data learning and integration, right? So that's our goal. So today I still need to, before, this week we focus on the protein structures, and I still need to spend one more lecture to talk about the concepts. Before, on Thursday, I will talk about the structured data. Okay, so bear with me, because there's still a lot of the concepts to be covered before we can address the structured data, okay? So the first concept is the secondary structure. You already have seen it, right? But I didn't really explain, right? How come you have a helix and you have a beta sheet, and what does this helix or beta strand or sheet means, and why do they matter? Okay. So the secondary structures are, due to the bonded and non-bonded interactions between the residues, right? You have seen these local three-dimensional structures, okay? So alpha helix, what we are looking at in a cartoon representation is like this. What we are showing is a cartoon representation of the backbone. Remember, the backbone does not consist of side chain. It only consists of the central carbon, amino group, and the carboxyl group, right? So this cartoon representation of the helix actually corresponds to a lot of atoms just interacting with each other through covalent bonds or non-bonded interactions. So bear with me. So I want you to focus on this first, this line. If you see this line going all the way, right? So remember, it's a one-dimensional chain of polymers. So these backbones actually trace in this line, okay? And you see that this line is sort of giving you a helix impression. Is that right? Right? So I'm going to show them in three-dimensional space in this figure, and at the end I will overlay these two views of cartoons and ball and sticks, okay? So let's look at the ball and the sticks, where each ball is an atom, or except the yellow one. Okay, let's forget about that for the moment. So I want you to pay attention to these black spheres, which are carbons. Okay, and let's look at these carbons, right? So, for instance, this carbon, right? So it also have different colors, like nitrogen, which is blue, and oxygen, that is red, and hydrogen is, oxygen is green, and hydrogen is red. It's not a typical choice. Usually you use red for oxygen for apparent reasons. Why? Because of the color of the blood. right? Oxygen is carried a lot in the blood and circulated through our body. So let's look at this as these atoms, right? So this one is, let's see, without a blocked view. Without a blocked view, so let's look at CO, okay. So this is a central carbon, right? So this is a central carbon. It's connected to a side chain, right? And then this is the CO, we are losing a OH, right? So this is a carboxyl group, right? And on it's also connecting to a hydrogen, right? And then the fourth one here is connecting to an amino group, right? So in other words, we see a single residue or a single amino acid, right? Now we can keep going. In this case, we have this, again, this amino group, right? Central carbon, and then this, the red one is a carboxyl group, CO, right? And then this is a hydrogen, right? So in other words, can you tell me which direction is the carboxyl group? Which direction is a carboxyl C end? Which direction is the amino N end, N terminus? Can you see that? Okay, we can leave that question later on, but you may wonder, okay, why isn't this chain, one-dimensional chain, be stretched out? What makes them forming this helix? Okay, you might notice that something, this kind of, these sticks, these solid sticks are covalent bonds, but we also have these, red, these dashed lines showing non-bonded interactions. It's exactly the non-bonded interactions makes this one-dimensional chain forming into a three-dimensional helix. Okay, let me give you one example. Right, this is oxygen, the green one, oxygen. This is on the carboxyl group of one residue, right? Then here, what you have is, the R group, and then there is a better view is here probably. Yeah, let's look at this oxygen, right? The green. It's forming interaction. Let me call it interaction for the moment. With a hydrogen attached to nitrogen. So this is the amino group, right? So in other words, the oxygen on the carboxyl group of residue I is interacting with the hydrogen on the amino group of residue J. That is a special interaction called hydrogen bond, remember, right? And the hydrogen bond, as we see, that is, contributing to a lot of the special properties like in the water because there are so many of them, right? So if you see that there are many of such hydrogen bonds, all right, along the axial direction of the alpha helix or along the chain. So those hydrogen bonds are the reason that this, one-dimensional chain is not stretched but actually forming a three-dimensional helix, okay? So now let's overlay these two views together. We have the half-translucent cartoon representation of the helix with the ball and stick representation of this helix together. And you can see clearly what we are showing in the cartoon is sort of, following the direction of the backbone, isn't it? Right? You see that, right? This is our backbone, right? And the reason that you have this helix is because of, these hydrogen bonds formed between residue I and residue J. And as I said, it's a carbonyl oxygen. That's the oxygen on the carboxyl group of residue I, and the amide proton. That's a hydrogen on the amino group of residue J. But there is a pattern. The difference between I and J is very regular. The difference between I and J is always 4. Okay? So because of that reason, you have a very patent alpha helix. So if you look on the top, you will see that every 360 degrees, like every turn, involves 3.6 residues. Okay, so that is the reason that you have a very patent alpha helix. Again, we see that hydrogen bonds is playing a major role forming this alpha helix. Okay. And this is another illustration, right? So again, this is probably a clearer view. In this case, we have the standard coloring where the oxygen is in red and nitrogen is still in blue, right? So this is central carbon, right? This is also numbered from 1 to 9. So this says that there are 9 amino acids starting from 1 to 9, okay? So for instance, here you have the C alpha of the first residue, and then you have, these COOH, that's the carbonyl group or the C terminus of the first residue, right? Then it's bonded with the second residue's amino group, this blue side nitrogen, right? Amino group. So this is the peptide bond connecting the two neighboring residues. So if you follow this, you see this nice helix. Okay. So this gives you a better view because I put the numbers there as well. Okay. These numbers are the index, right? Then you see that, for instance. This is the oxygen on the carboxyl group of residue 5, or so-called carbonyl oxygen on residue 5, is actually forming a hydrogen bond on with the hydrogen on the amino group of residue 9. So in this case, the hydrogen is in white. This is the more standard coloring, that oxygen is red and hydrogen is in light gray or white. Okay, so this is another illustration. And then if you remove, I was asking one question earlier. I was asking what's the direction of this protein chain? Which direction is N terminus? Which direction is C terminus? Can you see that? So it's from 1 to 9, right? So but even without the numbering, you see that, you start with amino group and end with the carboxyl group. Amino, carboxyl, amino, carboxyl, amino, carboxyl, right? And if you follow that direction, what you see is that the polarity in the one-dimensional space, or in this case, three-dimensional case, is going up from the first to the last residue. Is that right? Right? And then if you look at this one, what do you see? What does it remind you? When you learn, the electromagnetics, what you have learned is something called the right-hand rule, isn't it? This is actually a right-hand alpha helix, because if you do a right hand like this, your 4 fingers are following the direction of the backbone, and your thumb is following the direction of the N term to C term. Did you see that? Right? So it's an alpha helix, right? You may wonder whether there are other kind of helix. Yes, there are a lot of other kind of helix in nature, but there are reasons that they are less sampled by nature. Okay. So almost all helices found in proteins are right-handed. The reason is that what is not shown very clearly is the side chain. For instance, I never showed the side chains very clearly. This one threw away the side chain, right? In the previous figure, I showed the side chain as a single collapsed sphere, right? But remember, a side chain consists of multiple atoms, right? So if you want to form a nice backbone structure, you're going to make sure that the side chains, not only you have these attractions like hydrogen bonds between the backbone atoms, you also want to make sure that the side chains, which are big groups of atoms, don't clash or hinder the formation of such secondary structure, right? So that's the reason that in nature, you see most of almost all helices found in proteins are right-handed, because the steric restrictions imposed by the side chains in the left-handed helices, okay? And there are other right-hand sided helices like 310 and other things, but alpha is the most prevalent ones. And there are databases of the secondary structures such as the DSSP and so on, okay? So before I go to the secondary structure with the beta sheet, you may think about this. Secondary structure, what's the big deal of the secondary structures? To be honest, in the early days, when you want to make machine learning predictions for protein structures, this kind of secondary structure prediction is almost the first thing they started. Okay, I will talk about it later on. Why? Because it's not 3D prediction, actually. It's only 1D, right? Because every position you can say, oh, all these 9 residues are in alpha helix, right? Then maybe the next 5 residues are in beta sheets. So I can predict a sequence of secondary structures, which is 1D, okay, which is not that 3D. So it's in other words, it's like machine translation, isn't it? Right? You start with a text written in an alphabet of 20 letters, and you translate it into a text of secondary structures. Depending on how many secondary structures you have, if you have only alpha helix, beta sheets, and coils or loops, then you have an alphabet of 3 letters for secondary structures, right? Then it's like machine translation from one language to another, isn't it? Right? So there's no wonder that the secondary structure prediction was one of the first applications of machine learning in the world of protein structure prediction. Okay, is it still being done now? They are actually used more and more often as features or input to your machine learning models because they carry additional information. Okay. So the second, let's see, yeah. So the second, if you think about there are 3 major secondary structures, right? The last one is just the coils. Then the second one is a beta sheet. So usually we draw them like this kind of zigzagging sheets, and then they are like in parallel or anti-parallel in this case, right? With arrow directions, right? You probably figured out what those arrows are saying. It's basically showing the direction from N terminus to C terminus. Okay, but how could I draw a cartoon representation like this? What are the underlying 3D structures, right? Because eventually we want to predict the structures. So again, I use the same kind of illustration. What you see here is if you follow the black sticks, these are the covalent bonds that forming the whole backbone. You see these black sticks, right? These sticks are covalent bonds forming, connecting the amino acids. And if you look at the up and downs, or in the left and the rights, or bends and twists of these black sticks, it's essentially what this cartoon representation of backbone is showing. Is that right? So this cartoon representation is actually showing this, backbone structures, okay, in an ideal way. Okay, so it is indeed zigzagging, right? Right? Zigzag, 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 right? Just like what you see here, zigzag, 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 right? But what is underlying? Why wouldn't they form a nice one-dimensional thread, and they would rather form something like this in 3D space? Well, let's first look at, here you have multiple beta strand, and together the strands are anti-parallel and forming a sheet, okay? So let's talk about, apparently the reason is that there's something going on between the neighboring strands, right? Some neighboring strands that, instead of you have a first segment of the sequence going this way, but instead of keep going that direction, you would rather make a turn and then forming this, neighboring beta strand. Why is that? Again, I want you to pay attention to those, not the sticks anymore, which shows covalent bonds, but those dashed red lines, which are showing the non-covalent interactions. In this case, specifically, hydrogen bonds. What you see here is, again, sorry, there are two coloring schemes, but nitrogen is always yellow. I'm sorry, always blue. And in this case, nitrogen is connected to is bonded with hydrogen. Somehow it's in red in this case. And this hydrogen is making a hydrogen bond with an oxygen in the neighboring strand. So in other words, again, there are hydrogen bonds being formed between these, hydrogen in the amino group and this oxygen in the carboxyl group. But unlike the case of the alpha helix, where you see that these hydrogen bonds are being formed, along this whole single thread of the alpha helix. But in this case of beta sheet, you actually have the hydrogen bonds not between adjacent segments. Not within each segment. So, if you look at the alpha helix, right, within this segment, this is the alpha helix. Within this segment, we have the hydrogen bonds stabilizing the segment. But here we have the three segments, and the segments were put together anti-parallel because of the hydrogen bonds between the adjacent segments. So that's the difference between the alpha helix and beta sheet. They are both stabilized by hydrogen bonds, but in one case, it's stabilized by hydrogen bonds within the segment, that's our alpha helix. In the other case, it's stabilized by hydrogen bonds between adjacent segments. That's a beta strand, okay? What I didn't show here, if you go this way and then before you enter the next segment, there are some loop, very loose, non-stable secondary structures here as well, okay? I just didn't plot it. If you wonder, how come you have a broken protein, right? Actually, it's not broken. We just show the portion of the beta sheet, but if you start from the N-terminus, you go like, first, and then there's a loop, and then the second beta strand, then there's another loop, and then you enter the third beta strand, and then eventually go to the C-terminus, okay?
This bending is due to R group, not hydrogen group.
So that's a good point. I haven't talked about bending yet. I only talked about so far why the adjacent segments like to stick to each other because of the hydrogen bonds, right? But if someone asked a good question is, why are they zigzagging in this way? Why do they have to zigzag? It's a fantastic question. If you now overlay the translucent cartoon representation and the stick and ball representation of the protein, right? And ask yourself, how can I place the very bulky multi-atom side chain, these, currently represented like a yellow sphere, but remember, it consists of multiple atoms connected to each other, right? Some of them can be lengthy, right? So again, like what I said, why there are a few, left-handed helices in nature. Same here, that the conclusion was that, the side chains, right, being able to adopt side chains in 3D space without clashing them with each other or the backbone was a major reason of the dominance of the right-hand helices. Here, it's the same, that if you notice, this zigzagging leads to one pattern that is the orientation of the side chain. Here you see, it's like the side chains are always at the ridge of the zigzagging, and here it's pointing inside, here it's pointing outside, here it's pointing inside, here it's pointing outside, inside, alternatively, isn't it? So it's really a nice pattern, I don't know who figured this out. It's a really nice pattern because you want to use the building block of, building blocks of 20 amino acids and make sure that they can adopt different sequences and fold into different shapes and do different kind of functions, right? But how can you make that mechanism efficient, right? It's like a very sophisticated Lego, right? So in this case, this kind of secondary structure is in this shape because of the side chains is, is affecting the turns, in this case, the zigzag of the backbone. Okay, so that's the reason. Okay, and in this case, you probably noticed that I said before, you have to put the loops in between, and in this case, the neighborings are anti-parallel, right? But you could have this kind of parallel beta sheets as well. If you were to design these proteins, would you rather use more anti-parallel or parallel beta sheet, or do you think it doesn't matter? What do you think? If you are the designer of the proteins in nature, and you want to use these beta sheets, right? And you can arrange them in this anti-parallel way. The neighboring segments are anti-parallel, or this parallel way.
Maybe anti-parallel, because in that we have less unstable region, I guess.
Stability is one thing that you are hitting that is good, right? Yeah. So for instance, if you look at this one, this parallel case, did you notice that you have to make a very long turn? A very long turn that is occupying a lot of space, right? So stability and efficiency is one concern. Okay. There are more anti-parallel than parallel beta sheets, but it's not like the dominance of right-handed helices. Okay, there are still quite some anti-parallel. Quite some parallel beta sheets, because that's not the only concern. Yes, questions. Right. Good question. Did you notice that each segment has a direction? The arrow shows the direction that goes from N terminus to C terminus, the first letter of the protein sequence to the last letter of the protein sequence, right? So if you look at the arrow or the directions of the neighboring segments, in this case, it's anti-parallel, right? And in this case, it's parallel.
From the picture, it looks like helix.
It looks like helix to you?
Because the connection.
Oh, I see what you mean. Yeah, I think it's called a beta sheet because of the neighboring beta strands together form a very flat sheet. So that's, unlike the alpha helix. Okay. Yeah. Again, when we are talking about beta sheet, we are not looking at the turns. Okay. The turns are the third secondary structure, which are just turns or loops. Very unstable. Alpha helices and the beta sheets are more stable. Okay. All right. So now that we have learned so many concepts, I'm gonna ask you, how do we analyze this data? Imagine that this is the first time that protein structure is discovered, and you found even that there are some patterns of alpha helix and beta sheets, right? But you're a data scientist. How are you going to analyze? Are you going to design some algorithm or software to analyze the protein structures? The question is, how do you do that, right? Because so far what you learned is the concept or pattern by eyes, right? You learn this by eyes, right? The question is, well, how do we analyze this in a renewable way, right? So I wanna tell you a very simple algorithm, okay? A very simple algorithm. I think I like it because it shows you how do you do, dimensionality reduction in the real world, okay? Because this is a three-dimensional structure of thousands of atoms. Each atom has 3 degrees of freedom. And their collective pattern determines whether each of the residue is in an alpha helix or beta sheet or turn, right? And it's very hard for me to, translate that thousands times 3 coordinates into alpha, beta, or turns, right? If I want to see that, how do I analyze it? So that was the question, okay? So I want to show you a very simple algorithm. Okay, I wouldn't even, let's see what, so again, the goal is analysis. Here specifically it's a secondary structure analysis, okay? The question is, what are the patterns and why are there such patterns, okay? So as a reminder, right, so these are how peptides bonds are connecting the neighboring residues, right? Okay? So, something to keep in mind is that not all the not all the covalent bonds are equal in chemistry. Okay, so if you have this ball and sticks, and you can rotate these, for instance, I can fix this part and rotate the remaining by rotating around this bond. Okay, so in other words, these bonds are rotatable. Those are the torsional angles, but some bonds, due to the way the electrons are being shared, are less rotatable. So that includes the peptide bond. Okay. So if you want to analyze the secondary structure of every residue, what chemistry tells you is that, well, this is residue 1, this is residue 2. I should break, if I want to tell whether each residue is in alpha helix or beta sheets, I should break them here into different residues. This is what the chemistry would tell you. Is that right? Right? But that is actually not what this algorithm I'm going to tell you about is doing. Okay. So what another way, actually a more effective way, if you think about the thousands times 3 dimensions of degrees of freedom, right? Not all the degrees of freedoms are equal and they are coupled. So what another way of decomposing this polypeptide chain or protein is actually not at the peptide bond, but at the central carbon. Did you see these gray planes, right? Where they are connected with each other are at the central carbon, actually, C alpha, right? I have 1, 2, 3, again, 3 residues going this direction, okay? So actually, you can decompose this protein backbone using the so-called peptide unit, this covered by this gray, okay? So you may wonder, why do I do that, right? So it turns out that, these atoms within each peptide unit, as seen in this gray plane, are actually relatively rigid to each other, right? Because of the chemistry, they are not allowed to, move around with each other, with regard to each other too much, because of the peptide bond is usually regarded as not rotatable. Okay, so in this case, in other words, you have reduced the degrees of freedom because within each peptide units, there's no much relative degrees of freedom because they cannot move relative to each other. It's like a rigid piece, right? Then you can see that, each peptide unit is really like a rigid Lego piece now, and you can now connect the individual pieces or the individual peptide units, right? So the degrees of freedoms, what is remaining? When you are describing the degrees of freedoms for a lot of atoms connected with each other through bonded or non-bonded interactions, you can, of course, use thousands times 3 Cartesian coordinates. You can do that, right? But what is the difficulty? One, it's a lot of degrees of freedoms, right? It's a huge number. What is the second problem? There are a lot of constraints across the degrees of freedom, right? Because it's not like every atom, 3 degrees of freedom, can move freely relative to each other. If that's the case, we're not talking about protein, we're talking about gas, isn't it? Yeah. Okay? So in that case, if you are going to use thousands times 3 degrees of freedom in Cartesian coordinates, what you have is extremely high dimensional space, and in this space, you don't have very nice distribution. In other words, it's like, on the Pacific, there are a lot of scattered islands. Only on those islands, these degrees of freedoms are possible. But outside of the islands, most of the Pacific is not island, meaning that the combination of the degrees of freedom wouldn't be possible because they are violating the chemistry or the bonded or non-bonded interactions. Does that make sense to you? In other words, you have very high dimensional space with a lot of constraints. So an analogy is like, it's a on the Pacific, only some scattered regions are allowed. So if that's the case, it's really difficult for you to analyze. It's really difficult for you to search, right? So can you change the coordinates to something lower degrees and more contiguous? That's our goal, okay? So instead of using the Cartesian coordinates, you can use the internal coordinates like this, the length of the stick, that's a bond length, or the angle formed between 1, 2, 3 atoms, right, bond angles, and these dihedrals that are rotations around each backbone bond, okay? As I said, that this peptide bond here between the amino group of the previous residue, right, and the carboxyl group, between the carboxyl group of the previous residue and the amino group of the next residue, this peptide bond is not rotatable usually. So if that is the case, then if we think that the bond lengths are fixed, the bond angles are fixed, and the torsional angles involving 4 atoms are fixed for the peptide bonds, then the only remaining degrees of freedoms are what? Only the torsional angles around these bonds, right? One before C alpha and one after C alpha, isn't it? So in other words, what you end up having is, notice that this, I call it a phi or psi. That's, 2 means the index of the C alpha. Phi is the torsional angle around the bond right before the C alpha. And this psi is the torsional angle around the bond right after the C alpha. Again, because the direction is going 1, 2, 3, okay? From N terminus to C terminus. So specifically, phi is around the bond between nitrogen and the C alpha. So nitrogen and the C alpha. And the psi is between, is the dihedral around the C alpha and carbon, right? The C prime, right? That's the carbon in the carboxyl group, right? So now if we fix the bond lengths and the angles, then what we end up having is no longer thousands of degrees of freedoms that are all strongly coupled with each other. We only have what? 2 times L degrees of freedom left. Well, L is the length of the amino acid. Isn't it nice? Okay, so now let's look at this so-called Ramachandran plot. Okay, so in other words, we can look at the phi and the psi of every residue together and look at its pattern. Okay, so I don't want to talk too much details about it because this is high school geometry. Okay, but so that you understand the concept, I'm just going to say one thing, right? So again, this is phi angle, right? The phi angle is between the nitrogen of the residue I and the C alpha of residue I, okay? And to calculate this torsional angle, you are basically calculating calculating the angle formed by the 2 planes, right? This is atom 1, 2, 3. This is the plane formed by atom 2, 3, 4, right? Right? And atom 1 in this case is the carboxyl carbon of the previous residue, I-1, okay? So in the protein structure data, you have the 3 Cartesian coordinates of every atom, and you need to calculate these, the dihedral angle, okay? So this is high school geometry, right? So you just need to calculate, for instance, in this case, you can calculate the unit normal of these 3 of the plane formed by the atom 1, 2, 3, right? So this involves what? This involves cross products of 2 vectors, isn't it? Because you have this vector, line 1, and this vector, line 2. You want to calculate a unit normal, right, to the plane formed by these 2 lines that involves the cross product of these 2 vectors, right? And you can do the same to get the unit normal of the second plane, and then you can find out the angle formed between the 2. That is exactly this torsional angle phi, okay? So you may want to try some of this. I give some tutorials, not tutorials, just additional contents on the canvas, and we may do some simple homework on this. And be careful that the sign matters, okay? And similarly, you can calculate psi. So this is just the high school geometry, but in the end, so this is how you determine the sign. It's just some convention. I don't want to emphasize it too much. But in the end, what you end up having is you're looking at phi and psi, a two-dimensional visualization. Phi and psi only, okay? And you may have a lot of scattered plots or density distributions in this phi psi plot. So this is called the Ramachandran plot. So in other words, you are actually now visualizing the 3D structures with regard to its secondary structure pattern using a very straightforward, simple illustration. Because you have decomposed the protein into peptide units, you figure out that the degrees of freedom can be reduced down to phi and psi for each residue. So if I calculate the phi and psi of each residue and show the scattered plot or density distribution, you will actually find that some distributions, some regions of the phi psi distributions are corresponding to the alpha helix, some are corresponding to the beta sheets, and some of them are corresponding to the left-handed helices. Okay, so it's a very simple way. In fact, even right now, if you use AlphaFold, for instance, to make a powerful prediction, you still want to somehow do a sanity check. One of the sanity check is to see whether the distribution of the phi psi angles in the predicted structures are more or less agreeing with the distribution in the current data. Okay, so this is one way to analyze this. Okay, so this is a scattered plot. Okay, so I want to ask you a few questions. In this case, it's scatter plot. Okay, for instance, there's a homework. I can look at all the PDB structures or a subset of them. Right, I have to calculate the phi and psi for each residue of each structure, and each phi and psi pair of one residue in one structure is one dot, and then I show a scattered plot, for instance, in this case. Okay, but in this case, I show them in two different cases because there are 20 amino acids, right? 20 types of amino acids, right? So glycine is one of them. In this case, I'm showing the Ramachandran plot for all residues except glycine, so 19 amino acids, versus glycine. What do you see the difference? And, what's the difference in the pattern and why? First, we can talk about what's the difference, because, for all the observed, for all the residues except glycine, you see this, density distribution centered around alpha and beta regions and some small number of left helices. That is, according to our understanding, correct, right? But what do you see from the Ramachandran plot for glycines? What do you see? What's the pattern? Do you see a difference? What's the difference? No difference? So more regions in this phi psi plane are sampled in the case of glycine, isn't it? For instance, this region is represented a lot, right? Right? And this region as well, compared to the other 19 amino acids, isn't it? So in other words, glycines are more free to adopt all kinds of phi psi combination, aka backbone structure. Right? Why? Is that surprising? Any guess? Why? Why are glycines more free to adopt backbone structures? There's Rujia's answer is there's no side chain on the glycine. Yeah. So I think the answer is correct. But to make it more precise, glycine's side chain is the smallest. It only has one hydrogen. Okay? So in other words, glycine's side chain has the least restriction, right, on the structural choices of the backbone, right? So that's why the backbones are more free to adopt all kinds of combinations, right? Right? So maybe in your homework, when you do it by yourself and plot a Ramachandran plot for instance, prolines, you will see a very different distribution as well, and you may ask why. Okay, so that's one kind of, another kind of exercise. Yes, question?
So I'm a little bit confused. So like the three secondary structures that we learned,
Right.
like they're like always either like beta sheets or like alpha helix or the loops, or like there are other like the structures as well, but
Yeah.
not prevalent.
Yeah, very good point. So usually we classify secondary structures into 3 classes or 8 classes. So if you classify them into 3 classes, then it's just alpha, helix, beta sheets, and the loops. But if you want to, look into them more carefully and show more higher resolution, then you may consider 8 classes. For instance, the 310 helix is one of the 8. Okay? So in other words, it's just how many classes of the label you are adopting. It could be 3, it could be 10. Or what's the size of the alphabet for the secondary structure text? Okay, good question. Any other questions? Okay, so now we talked about a sequence. We talked about one analysis algorithm, right? So simple, but it's really not that easy to figure out. Okay, so that's why it's named after the scientist Ramachandran. Okay. And it's still used very often, even in the post-AlphaFold era. So now let me show a second algorithm. I'm not showing an algorithm. Actually, I'm showing a second utility of the algorithm. That is a prediction. The question, usually the prediction kind of task is asking what would happen given this, okay? So our question is, given the one-dimensional sequence of the amino acid, what are the secondary structures of each position? Okay, which portion is alpha, which portion is beta, and so on. So that's a secondary structure prediction question, right? So as I said, that this is one of the earliest machine learning problem. And if you don't know who Martin Karplus is, I regard him as my academic grandfather. Anyway, so also a Nobel laureate at 2013 in chemistry. I just want to remind you that Martin Karplus already used the neural network in 1989, probably before you were born, to predict the secondary structure, okay? So neural network is nothing new, okay? But deep learning is slightly different. Anyway, and also back then there were not a lot of data. There were not enough computing resources. But anyway, I mean, these are the founding fathers of this field. But anyway, one of the elementary tasks, prediction tasks, still remain in secondary structure, in the structure of bioinformatics. That is to predict the secondary structure. Okay, I just said that I think now with the advancing of data, right? We have way more sequences already, and we have more sequences that are labeled with 3-class or 8-class secondary structures. This question can be answered much more effectively now than 1989, right? So I mentioned the machine translation formulation. I think that is one way, right? So you can use recurrent neural networks, for instance, right? And machine translation, I think you can use other things as well, right? So one of the recent papers are actually using this RNN framework to start with a one-dimensional sequence and predict this, the secondary structure sequence. So it's a sequence-to-sequence translation formulation. It's still relatively easy. And we'll briefly revisit the topics when we go to the data-driven approach. And there are some review that you can look at. Now, what is the secondary structure prediction problem? The input is a sequence. It's a text written in alphabet of 20 letters, but sometimes you can see a format for the sequence called a FASTA format. So what is a FASTA format? Actually, you can just regard it as, something right before the actual text. You have a header. You have a header starting with bigger than this symbol. Okay, so this right before the line of sequence data, you have a single line description header, and you can determine its own format. For instance, this is example 1. This example 1, this FASTA sequence is taken from the NCBI entrance. So you see that they have a lot of vertical lines separating some keywords. So these keywords can be used to cross-reference different data sets. Okay? For instance, in this case, it can include the entrance gene ID, protein ID, and the UniProt ID. So these are different data sets, and these are the different IDs so that you can cross-reference. Okay? And in the case of the protein data bank, we'll talk more about it next time. You see that these vertical lines are separating the 4-letter code of the PDB ID, and then colon, and then the chain ID, and then this PDB ID chain sequence. So this is kind of the header. You can decide what is the format, but usually that is where you can put the additional information. Okay, we recently designed some sequences, and then we want to still convert them into FASTA format, but we didn't really need any information much information in the header, so we just wrote them as bigger than designed by what model with what setting, and then with what filter, for instance. Okay, so this is the FASTA sequence. The output, as I said, is because our labels in this case are discrete, right? It's 3 classes or 8 classes. So it's a classification problem, right? Your labels are discrete, so it's a classification problem. So in the early days, they are really solved, shallow machine learning models using classification, right? So for instance, this is one server called Raptor, right? So you see that the first line is the sequence, PIVKRLLGWK, right? Proline, isoleucine, valine, lysine, arginine, leucine, and so on. Okay, glycine, and so on. And there's position 1, position 11, and so on. Okay, so the first row is the input, that is a sequence of the protein. And the next 4 rows are the predicted secondary structure and other labels. Okay. For instance, this is SS3, meaning secondary structure, 3-class secondary structure. So Nami was asking about whether you have 3 classes or more classes, right? So you see that in this case, we predict these labels in both 3-class and 8-class, okay, secondary structures. So we see that in the case of the 3-class secondary structures, position 1 is a proline, it's predicted to be in the C, that's a coil, okay? Position IVKRLLG is predicted to form a helix segment, H, okay? Then when you see an E, that means it's a beta strand, okay? And similarly, you can look at the secondary structure in the 8 classes, okay? And you can also predict other things that are not secondary structure, but related to the structure. For instance, you can predict which position or residue is hidden in the core of the protein and which are closer to the surface of the protein. So in other words, you can predict the context. So in this case, E or M are showing whether it's and B, I think they just show that whether it's buried or in the surface or somewhere in between. Okay, so there are 3 kinds of labels showing where they are from the core to the surface of the protein. And lastly, you are predicting which positions are disordered, because so far I'm showing that everything is nicely ordered, but the reality is that in our bodies there are a lot of proteins that are not structured. They are intrinsically disordered. You may wonder, well, you told me that a protein needs to adapt into a 3-dimensional shape to function. Now you're telling me that a lot of proteins don't have structures. So what are you doing? Well, it's still a ongoing study, and there are a lot of discussions, but even about what is the definition of intrinsic disorder proteins. But one theory is that if you have a protein with some intrinsically disordered regions, then that region is actually quite flexible to do different things in different environments or contexts. Okay, so that's one of the theory. So lastly, I want to talk about the tertiary structures. Now that you have learned the secondary structures, local secondary structures, right? And you see that they are classified in 3 or 8 types, and why they can be treated as a classification problem, and intrinsically it's a 1-dimensional sequence as well. So that's why you can do this classification as if you are doing the translation, right? Like translating one to another. Actually, it's easier than translating from English to French. Why? Because there's a 1-to-1 correspondence between the characters. It's actually much easier, okay? So if you're thinking that, oh, some of the most advanced human language models for translation needed to be done in that way, you have to ask yourself how much of that is needed for our case, and how much of our special properties can be utilized better in your new algorithms. Okay, so that's something to think about. That's what I mean. AI is targeting these applications, but it's also inspired from these applications. So lastly, the tertiary structures. So the tertiary structure is usually the global 3D structure, right? But there are 2 things, still 2 concepts I want you to be aware of. One is what is a domain, and one is what is a fold, right? So each single tertiary structure corresponds to 1 chain, and then it can form spatially separate regions, and they are called domains. I will show you later on an example. So these domains are spatially far away from each other, relatively stable and well folded, but there are also conserved parts that can evolve, function, and exist independently. Okay, and then depending on within each domain, depending on how the secondary structures are arranged relative to each other, the pattern is called a fold. Okay, so I'll give you a visualization. This is one protein called a pyruvate kinase. What it's doing is catalyze the following chemical reaction. Right? That is, it's an enzyme. It's a pyruvate kinase. It's an enzyme that catalyzes the following chemical reaction, right, to convert the reactants to the products in this case, right, with the conversion of ADP to ATP, right. So if you ask what is this protein, right, this protein actually, if you look at this, the code is 1PKN. I show the backbone cartoon and then already color them in 3 colors. So you can sort of see that there are 3 subcomponents of this 3-dimensional shape that are relatively, separate from each other, and each of them is packed, right? You have very packed individual domains that are relatively loosely connected with each other, like here. Okay, you have this, right? And for each domain, there are some pattern of how alpha helix or beta sheets are packed to each other. That's called the fold. Okay. So it's very fascinating. If you look at the top domain, this is actually where the protein binds to the nucleotides. Okay, so and the secondary structures are mostly beta sheets. You don't have alpha helix here, so it's an all beta kind of fold. Okay, and the central domain, the gray one, is actually binding to the substrate and helping this chemical reaction. Okay, and it actually contains both alpha helix and beta sheets, and the alpha and the beta are alternating. Okay, one alpha, one beta, one alpha, one beta strand. And the beta strands are actually mostly parallel because they are separated by the alpha helices. So this is a alpha slash beta, alternating alpha beta kind of fold. And the bottom domain is more for regulatory functions. And again, it's alternating alpha and beta, okay? So you see that there are three domains, right? They are, structurally separate. They are relatively compact within each other and structurally separate, but they are actually performing different functions. Actually, if you wonder how nature made this protein, actually each of these domains might have evolved independently already, and then they're put together, assembled together somehow. Okay, so that's amazing part. I want you to pay attention to one thing. I'm not saying that from N to C terminus, if you follow the sequence, you go through the domain 1, then domain 2, and domain 3, and then that's it. No, if you see here, you see that you're entering domain 3, and then you're finishing domain 3, and you're entering back to domain 1. So in three-dimensional space, they are separated from each other relatively easy. But in one-dimensional sequence, you may see domain 1, domain 2, domain 1, domain 2, then domain 3, then domain 2 again. You see what I mean? So that's a little bit complicating, okay? So in other words, the domains are compact in 3D, but not contiguous in 1D, okay? So, if you look at what these patterns, how these, alpha or beta secondary structures are packed together, and each pattern is like a fold. I'm just giving you one example, one fold example for each category. In the case of all alpha, you have this globin fold, right? In the case of all beta, you see this is top view, which is like a, called a beta propeller, a very nice very nice structure. You can have this all beta structure forming this beta propeller. And this is a team barrel. You have the top view and the top view and the side view. It consists of alternating alpha beta. For instance, if you follow this, let's see. If you follow this N terminus, you go through a beta first. Then you come back, you see alpha. Then you follow through the next one is beta, and so on. Okay, I'm not counting the loop, but the stable secondary structures are alternating between alpha and beta. You could have alpha plus beta, for instance. In this case, start with usually this. We are using this rainbow coloring. So the blue is N terminus, and the red is a C terminus. Okay, so coldest is N terminus. So if we follow in the order, we go through beta, alpha, beta, beta, alpha, beta. So it's not alternating alpha beta anymore because we had this, two betas already neighboring each other, forming a sheet. So in this case, it's alpha plus beta, okay? So each individual fold has this three-dimensional pattern, and it's also very often related to a function or some functions, okay? So in the end, let's summarize and see whether there's any questions. Today we still talked about a little bit more concepts about the structures so that we can understand the tasks and the algorithms, okay? So we talked about the secondary structures, mostly the three-class definition. We talked about what made alpha helices and beta sheets because of the hydrogen bonds within a segment or across adjacent segments that form alpha helices and beta strands, sheets, respectively, right? We talked about